Hello everyone. Today we are going to study about subviral particles. First of all, we'll know about viroids. Viroids solely consist of small single-stranded circular RNA having molecular length of 250 to 370 nucleotides. It was Theodore Diener who was studying on some plants, and he discovered acellular particles that he named as viroids, meaning virus-like. And these viroids doesn't code any gene products, and they also they cannot replicate themselves, and they depend on host RNA polymerase 2, which actually synthesizes a negative strand RNA, and then this negative strand RNA serves as template for synthesis of other viroid RNAs. And there is no code protein as such. It's just simple single stranded circular RNA which is naked. This is the mode of replication of viroids. Viroid genome gets replicated through rolling circle mechanism using host polymerase 2, and a linear concatemer of viroid genome is formed, right? And then because of self cleavage activity, it gets converted into linear antigenomic RNA. Antigenomic means a copy of the genome, and the sense of this strand is a negative sense. And then this linear antigenomic RNA gets circularized and again get replicated through rolling circle mechanism by host polymerase 2, okay, RNA polymerase 2. And again a concatemer is formed that undergoes self cleavage and converts into linear genomic RNA, and upon circularization a viroid genome is ready. So this way viroids get replicated. Now there are several modes of transmission of these viroids. One of them is through mechanical damage that can occur during horticultural or agricultural practices, and also they can be transmitted by aphids as well as from plant to plant by leaf contact. There are several diseases caused by viroids. There are about 20 diseases. Few of them are potato spindle tuber disease (PSTV), chrysanthemum stunt disease, that is CSV, that cause some malformations in tubers and leaves, right? And there are other examples of these viruses like tomato plant mycoviroid, which infects tomato plants and cause loss of chlorophyll, and it makes leaves brittle and disfigured. The plant will grow very small tomatoes, and which results ultimately in loss of productivity in the field crop. And avocado sunblotch viroid results in lower yields and poor quality fruits. You can see in this image that the fruit quality of the avocado is really degraded. And this viroid particularly, is the smallest viroid discovered so far that infects plants. Another viroid is peach latent mosaic virus that can cause necrosis of the flower buds as well as branches, and wounding in the ripened fruit, which ultimately results in fungal infections and bacterial infections in the fruit. This can also cause similar pathological changes in plums, nectarines, apricots, and cherries, resulting in discrete changes and decreased productivity in these orchards of these fruits. Now, another term that I want to introduce you to is virusoids. Virusoids are circular single-stranded RNA molecules which depends on plant viruses for their replication and encapsulation. The genome of plant virusoids consists of several hundreds of nucleotides, and it doesn't code any protein. Now, a viroid that infects animal cell is HDV. HDV viroid is encapsulated in the hepatitis B virus, and it is a negative sense single-stranded closed circular RNA genome having viroid. And this ssRNA genome of this viroid consists of 1700 nucleotides, and it can direct the biosynthesis of some HDV associated proteins. Now, infection with these viroid results in liver failure and rapid progression of the liver cirrhosis. In combination to HBV, HDV has a highest mortality rate of all the hepatitis infections known. This enters in the bloodstream and can be transmitted via blood or serum. Now, next subviral particle that we want to discuss is prions. Prions are infectious proteins. Uh, this These are proteins. Okay. Earlier subviral particles that we studied were nucleic acids. Prions are proteins. It was Stanley Prusner, who was a medical doctor studying scrapie, who discovered the disease scrapie, which was caused by proteinaceous infectious particle, which are known as prions. Prions are actually primarily consist of sialoglycoproteins. So you know now that these are glycoproteins, and these are known as PRP, prion proteins. These contain no nucleic acid. Prions are known to cause several forms of transmissible spongiformis encephalopathies in humans as well as in human animals. Stanley Prusner received Nobel Prize in Physiology or Medicine in 1997 for his contributions. And this slide is showing you the normal prion protein and altered prion protein, which is infectious in nature. So this normal prion protein is rich in alpha helical structure, while this infectious prion is rich in beta structures. Right, and accumulation of these rogue proteins causes the brain tissue to become sponge-like, spongy. It's spongy for me, right? And killing of brain cells results in formation of holes in the tissue, leading to brain damage, loss of motor coordination, and finally dementia. You see, a normal endogenous prion protein once comes in contact with the infectious prion protein get transformed into an infectious protein itself. And these infectious proteins together accumulate in the neuronal tissue and causes the killing of the brain cells and formation of holes in the tissue. These solid dark markings are of these aggregates which are causing the damage of the brain tissue. There can be different ways these diseases can transmit or can be acquired. So first is the acquired way of disease, then familial and sporadic. And currently research suggests that the primary method of infection in animals is through ingestion, and there is no evidence that it transmits via casual contact with an infected person or animal. Now in cattle, we know that mad cow disease, scrapie, and chronic wasting diseases are caused by prions. In case of humans, Alzheimer's disease, fatal familial insomnia, Kuru disease are caused by prions. There are other examples in humans, including this GSS, that is Gerstmann-Straussler-Schnittger disease, and Creutzfeldt-Jakob disease, CJD, right? So in humans, there are different mechanisms of transmission: sporadic, variant, familial, iatrogenic. Okay, so these are like the reason for sporadic CJD is not known, and it is thought that some somatic mutation may cause this disease, resulting in alteration of normal prion protein. While variant CJD may be resulting from eating contaminated cattle products or by secondary bloodborne transmission. Familial CJD results because of mutation in germline PRP genes, and iatrogenic CJD results because of the contaminated neurosurgical instruments, corneograms, gonadotrophic hormones, and secondarily by blood transfusions. Kuru disease results because of the eating of infected meat through some ritualistic cannibalism, and GSS results because of the mutation in the germline PRP gene, as well as fatal familial insomnia also results from the mutation in germline PRP gene. Okay. Now, one more thing we should know is that prion destruction is really difficult because they are resistant to heat, chemicals, and radiations. Even standard sterilization process may not work for destruction of these particles. So, and because there is no cure of TSC diseases, so utmost care should be taken, and for guidelines should be followed handling the meat and infected animals. So so that we can prevent the transmission of these diseases. Now the final or the last subviral particle that we are going to study is virusoids. As I already told you that virusoids are subviral particles best described as non-self-replicating single-stranded RNA molecules which require help of other viruses to replicate and to encapsulate themselves. Right, so they are dependent on helper viruses. Their genomes are small and they do not code for any proteins, and usually the helper viruses for virusoids comes from the family Sobemoviruses. Okay, and these helper viruses, unlike virusoid, undergo normal mode of replication, whether and they are not dependent on any other viral subviral particle or virusoids. Like virusoids are dependent on helper viruses, but helper viruses are not dependent on virusoids. So once virusoids and helper viruses both are in the host cells, helper virus facilitate the transcription of the virusoid genetic material. So this way we end our present lecture on subviral particles, and thank you for watching this video. Keep coming to this channel for more wonderful updates. Thank you.